Hello, everyone. Welcome to week number 5. We are past the halfway point in the term. So if you're still here, if you stuck it out with me this long, you don't have very many weeks left of this science class before you can move on and take something else. So, well, we have more science to do, so let's take a look at that. All right. Last week, you learned about scientific communication, whether it's communication between scientists, groups of scientists, you and your colleagues, you and other professionals, or scientists in the general public. And you have to figure out who your audience is, who you're speaking to, who you're presenting to, in order to effectively communicate with that person or those people. So determining your audience is terribly important, and you had to do that for your project too. You decided which group of people you would be presenting to and then how you would tailor your presentation to that group. How much explaining you would have to do and how much you wouldn't have to do. So that's very important. This week is all about biases in science and also probabilities, because we see a lot of statistical work in scientific reports and scientific articles and scientific research. And so you need to know what that means. So let me go through some of this. Biases in science. We try to take out bias. What does bias mean? It means that you come with a certain viewpoint and you want to get that viewpoint across, but that's not how science works. Science takes out bias. You just look at the results and determine what that means. So what kinds of bias are there? The researcher brings bias because the researcher has expertise in this subject. Yes, he or she's developed these experiments that will be done, but you have a certain expectation about what is going to happen because you understand this. Does that always happen though? Do you always get the expected results? Absolutely not. And some of the greatest strides that have ever been taken in science have had this happen, where the unexpected happens and then you have to figure out what that means. So it's really important to take out that bias. Your client, your patients, your subjects, whoever it is that may be in your particular study also come with biases, and they want to help you, and so they want to give you the answers that you want. So you have to take that out too. Also, if a company is paying for your research, don't you think that company has an expectation that you're going to give them the results that they want? Absolutely. Should you take that out? Yes, you should. You get what you get, and that's all there is to it. I'll give you a quick example of this. Back in the late 1990s, I was an expert in DNA fingerprinting then, and there was a lawyer in Greenville, South Carolina, who was representing an alleged father in a paternity case. And this was way back at the beginning of DNA fingerprinting. And so it was really rough. There were not even set guidelines on how you did the technique. And so he showed me an X-ray of DNA from mother, child, alleged father, but nothing was labeled. Nothing. So if you turned it the other way, then you got a totally different result. So I said, I can't read this because nothing's labeled. I can't make sense of it. And I said, do you have any other results? And he said, yes, they did quite a few blood typing tests. The ABO blood group and then the RH factor plus or minus, right? Those. Well, there are lots of other ones. Well, they had done 10 different blood typing tests on this trio, mother, child, and alleged father. And I said, well, let me take a look at those. So I did. And I sat down. If you've ever done a Punnett square in genetics before, you know what I did. I sat down and I worked out Punnett squares. And I went back to the lawyer and I said, well, I've got some news for you. None of these tests rules out your fellow as the father. And that virtually means that he is the father. It's not 100%, but there was not a single one of these different 10 blood typing tests that I worked through that would eliminate him as being the father. So you see, I had to tell the lawyer exactly what I found, even though that wasn't what he wanted to hear. And the fellow started paying child support payments to the mother. So anyway, that's an example of that. I did want to mention that there are double-blind studies, and this means that the researcher doesn't know what the treatment is or whether it's a placebo, and neither does the client, patient, subject, whatever you want to call them. This is what happened with me with the Pfizer vaccine clinical trial that I participated in. I was given the placebo and the booster of the placebo. My doctor didn't know that, and I didn't know that. But when it came out, after they had looked at all the data, they actually went back up, opened up the double-blind study, and gave those of us who had gotten placebos, they gave us the actual vaccines and then the boosters. So that was kind of interesting. I was in the placebo group. All right, then we talk about p-values a lot, because you want to know what the probability is that this occurrence happened just by chance. So if you see a p-value that's less than 5%, 0.05, it would be the p-value, then that means that there is very little likelihood that this happened by chance. There's only about a 5% chance that whatever this connection is that you have made happened by chance, that it really was the effect of whatever the thing was in the experiment. Confidence levels are sort of the opposite of that, like 95% confidence that this effect is true and not happens by chance. So that's sort of the opposite of the p-values. The chapter in the Sumo web text talks about Gregor Mendel, who was basically the father of genetics. He was an Austrian monk that did a lot of experimentation with pea plants. He grew thousands of plants and got even more thousands of seeds, and then planted those and figured out how traits were passed along through the parent plants. And he came up with the terms, which you know, dominant and recessive traits, right? He came up with that because he could figure out how traits were passed along through the generations. So, and his experiments were very simple. And peas, just like they are today, are a food crop, and so they were readily available in different varieties. So he had a simple type of experiment that he did in order to understand genetics. The most important contribution of Gregor Mendel is the fact that he counted and characterized every one of these offspring. And because he counted, he could figure out the relationships, which is really, really cool. The fact that if you have a dominant trait, it is expressed no matter how many copies of that trait are there. If you have a recessive trait that's being expressed, that means that there are 2 copies of it and that there is no other dominant trait present. See, we know that already, and that comes from Gregor Mendel. It shows you how science is done and how you can design a really good experiment, and it doesn't have to be really big and complicated and expensive. You'll delve a little bit further into the Deepwater Horizon oil spill in 2010 and the importance of the scientific examination of the flow that was coming out of the oil well in the ocean. The company was saying it was only about a tenth, I think it was, of what the scientists came up with, and this made the world of difference in how they were going to have to stop the spill from happening, because at that point the oil was just still flowing out of this well, and so it was still polluting and polluting and polluting and polluting, and they had to figure out a way to stop it. And the only way that it was stopped was by the scientific examination of this, because of course the company is going to downplay this, right? I mean, that's the way that the world works. The company was downplaying how much oil was coming out, and because they did, there wasn't a way to cap it off, which they ended up not doing. They ended up drilling another well to alleviate the pressure, and then they could cap off the one that had ruptured. So that's a very interesting read right there. All right, so what do you have to do this week? You have your rough draft and a new section called the hypothesis section. You should take a look at my feedback from your rough draft from week 4 and make those corrections as you need to, but you're also going to add a hypothesis section. And you may think, I've already developed a question way back at the beginning, the second week. Okay, so I want to talk about that for a second, because yes, you already formulated a research question, some part of this topic that you have chosen that you wanted to look at, but now you have to make a hypothesis. And a hypothesis is usually in the form of an if-then statement. I'll get it out in a second. If-then statement. If this happens, then that's going to happen, okay? But what's the difference between your question and your hypothesis? Your research question was an overarching question that you wanted to delve into, that you wanted to examine by looking at the literature. What's the environmental cost of using lithium batteries in electric vehicles? How does ocean acidification affect marine life? You see how huge these questions are? Okay. They aren't really testable, are they? You got to get down to the nitty-gritty to ask a question where then you can design an experiment. So that's what your hypothesis is. It is a more specific question that is answerable by experimentation. So you got to get way down on the experiment level in order to develop a hypothesis. And right off the top of my head, I can't think of one right now, but you could choose oyster beds, and you could bring them into the lab, oysters into the lab, and you could grow them at different pHs, and you could see what happens to them, what happens to their growth, what happens to their reproduction. There's all kinds of ways that you could look at that, right, if you're interested in ocean acidification. So if the pH of ocean water, and you can simulate it, is lowered, then oysters will produce less offspring. That's a hypothesis that you could develop experiments on. Much different from what is the effect of ocean acidification on marine life, right? That's a huge question. A hypothesis is much more specific. So that's the difference there. Make sure you use your rubric to guide your work, and the rubric is in the web text, and it's also in your guidelines and rubric that's in your learning module also. So don't forget that. As always, if you have any questions, please let me know, because I can help you, I promise. All right, tip of the week, use the feedback that you get from me in order to improve your work, because in week 6, when you turn in your final copy of your written paper for your project, that's it, and it's a really big grade. Take a look at how many points it's worth. It's worth a whole lot more points than any of these smaller assignments. So you want to fix everything by the time you get there to week number 6. All right, so please look at your feedback from me and improve your work. That's very important. There's my name again, Dr. Janie Sigmon, m.sigmon@snhu.edu. Just let me know if you need anything. I'm here for you. I'll be glad to answer questions or to help you. That being said, I hope that you have a great week number 5.